Bismillahirrahmanirrahim. Omics Peak Part 2. Transcriptomics. Transcriptomics is the study of all the transcripts that are present at any time in the cell. In principle, the transcriptome includes messenger RNAs, mRNAs, in addition to ribosomal RNAs, rRNAs, transfer RNAs, tRNAs, and small nuclear RNAs. But transcriptomics is usually limited to mRNA, the template for translation into protein. The main activity in transcriptomics is to obtain a profile of global gene expression in relation to some conditions of interest, which genes are turned on and off during certain phases of cell cycle, which genes are upregulated by certain physiological conditions, which genes change their expression in response to adaptation to the environment. The study of transcriptomes is part of functional genomics because it does not look at the DNA as such, but at its functions. In general, it is expected that there are more transcripts that there are protein encoding genes in the genome, even when considering only those genes that are actually transcribed. This is due to the mechanism of alternative splicing, the generation of different mRNAs from the same pre-mRNAs during the removal of introns, RNA editing, post-transcriptional insertion or deletion of nucleotides, or conversion of one base for another is another reason for incongruence between the genome and the transcriptome. Classical molecular phylogenetics, Grohr and Lee 2000, is based on variation of homologous DNA sequence across species. However, the same structural DNA can be regulated in different ways in different species. We illustrate this argument with an example from Enard et al. 2002, who did one of the first studies in comparative transcriptomics. Enard et al. 2002 analyzed the expression of 18,000 genes in the liver, blood, leukocytes, and brain tissue of humans, chimpanzees, pan troglodytes, and rhesus monkey, Macaca mulatta. Chimp brain, rhesus, human, blood 5.5, chimp 1.0, human, rhesus, and chimp 1.3, human, liver, rhesus. Bismillahirrahmanirrahim. Omics Peak Part 3. Proteomics. Proteomics is the study of all the proteins in the cell. Mass spectrometry may also be used to determine the amino acid sequence of a protein. For this application, the protein is cleaved with the protease, for example trypsin, which generates a collection of fragments characteristic of the protein. These fragments may be compared to an in silico computer simulated digestion derived from the genome and the known cleavage sites of the protease. The proteome provides a different picture of a cell's activities to the transcriptome. Several authors have indeed wondered about the lack of correlation between mRNA and protein abundances. One of the reasons of this is the existence of control mechanisms at the ribosomes, where mRNA is translated to peptides. Translational control allows the cell to select only certain mRNAs for translation and block others. The selection is often dependent on environmental conditions, so this mechanism allows for physiological adaptation on the level of proteome. even though the transcriptome remains the same. Another issue is post-translational modification or protein processing process that can greatly affect the function of a protein, for example, by acetylation or ubiquitination of the N-terminal residue, hydroxylation of prolines, or cleavage of the molecules into smaller units. The proteome and the genome are linked by many feedback mechanisms because some proteins are transcription factors necessary for gene activation, others are enzymes involved in transcription or translation, and still others are structural components of chromosomes. So, in a molecular biology context, the living cell can only be understood fully by considering genome, transcriptome, and proteome together. Omnics Speak Part 4. Metabolomics. Metabolomics is the study of all low molecular weight cellular constituents, usually only metabolites belonging to a limited category are included, for example, all soluble carbohydrates or all metabolites that can be measured by a certain analytical technique such as pyrolysis, gas chromatography, or infrared spectrometry. No single method can measure the thousands of different chemical compounds that may be present at any time in a cell. Because of the greatly diverging chemical properties, hydrophilic versus hydrophobic compounds, acids versus bases, reactive versus inert compounds, etc., the metabolome requires a diversity of analytical approaches to obtain a complete picture. List of some of the more common omics term and object of study. Pathogenomics, genomes of human pathogens, analysis of genes involved in disease generation. Pharmacogenomics, genomic responses to drugs, analysis of expression profiles that indicate similarity of action across compounds. Analysis of genetic polymorphisms that determine a person's disposition to drug action. Toxicogenomics, mode of action of toxic compounds, development of expression profiles that indicate similarity of toxic action across compounds. Ecotoxicogenomics, genomic responses of organisms exposed to environmental pollution. Ionomics, all mineral, nutrients, and trace elements in an organism, for example, using inductively coupled plasma mass spectrometry.